All right, welcome to the lectures on epithelial tissue. So overall, the body is comprised of four different tissues. Tissues are just groups of cells working together for a common function. We call the study of tissues histology. It's a type of microscopic anatomy. And the four different types of tissues are epithelial, connective, muscular, and nervous. In class, we're probably going to go over epithelial and connective, but separately, we're going to talk about muscle, and then we're going to spend quite a lot of time talking about nervous tissue in separate lectures. So epithelial tissue, also called epithelium, is made out of one or more layers of closely packed cells. To go backwards here just for a second, muscle tissue gets short. It contracts. That's what it does. That's its thing. Nervous tissue processes and sends electrical signals. Epithelial tissue does all of the absorption and secretion in the body. And then connective tissue just kind of holds everything in place. So let's talk about the epithelia. Again, one or more layers of packed cells. Really isn't a lot of cellular matrix. It's just cells. There really isn't a lot of stuff around them. It covers your body surface, it lines body cavities, and makes up the majority of the glands in your body. So those closely packed cells are held together typically with tight junctions and desmosomes. They have a polarity or a direction to them. There's an apical surface, which is exposed to an external environment or an internal environment in the case of a body space. In other words, they'll face a lumen. And sometimes the apical surface will have microvilli or cilia on them, but they don't have to. And then the basal surface usually sits on some kind of connective tissue that's holding it up and supporting it. The epithelial tissue attaches to the basement membrane. There's three layers. There's a lamina lucida, a lamina densa, and a reticular lamina. There's a lot of collagen, glycoproteins, and proteoglycans, and these are all important, especially in connective tissue, to help hold it together and help the epithelial cells attach to that connective tissue. Now, one thing to note here is that epithelial tissue is avascular. In other words, blood doesn't flow into epithelial tissue, it flows near it. So the nutrients, rather than taking the bloodstream into the epithelial tissue, get right next to it, and then we have to start using diffusion to move oxygen and glucose and other nutrients into the epithelial tissue and to move waste products back out of the epithelial tissue to where it can get into the connective tissue, which is then vascular. This leads us to kind of a problem I like to call the pancake conundrum. If you have a stack of pancakes, and you pour syrup on that big stack of pancakes, the syrup doesn't get all the way to the middle of the pancakes. It's too thick. So in other words, the syrup can't diffuse across a large distance. And so we have this problem with epithelial tissue where if it's too thick, then the nutrients can't diffuse to it in time, and that epithelial tissue is going to die. So when we look at epithelial tissue, because it is avascular, it tends to be fairly thin in order to survive. It is extensively innervated, though. There's lots and lots of nerves that run into epithelial tissue, and it has a crazy high rate of regeneration. Some of the epithelial tissue cells, for example, in the small intestine, will turn over every 3 days because it's such a harsh and unhappy environment. So here's an example of some epithelial tissue. We've got our apical surface up at the top. Now this is going to face a lumen or outside the body. We've got our basilar surface here that's sitting on a basement membrane with connective tissue underneath of it. You can see our connective tissue right here. And then here is our layer of epithelial cells. In this case, there's only one layer. You can see, but if you count the nuclei, kind of go across there, there's only one layer of nuclei, so that will help you to count how many layers there are. So epithelial tissue can be used for lots of things. We use it for physical protection. It helps protect us against dehydration and abrasion. It helps protect us against infection. It's selectively permeable, so some substances can get through the epithelial tissue, some cannot. It functions in secretion. Secretion is when a cell produces a substance and then pushes it out of the cell, typically through exocytosis. So our glands do lots and lots and lots of secretion. Excretion is when the body produces something and that leaves the body. So in other words, when you defecate, you are excreting. When you produce tears, they are secreted from your tear gland, move through tear ducts, and then if they leave your eyeballs, then those tears have been excreted onto your cheeks. Epithelial tissue also has lots of nerve endings, so it's great at picking up sensations, things like touch and pressure, temperature, and pain. And then we also have some epithelial that's very specialized. We call that neuroepithelium, and it houses cells that are responsible for sight, taste, smell, hearing, and equilibrium. Now, when we look at epithelia, we like to classify it, one, by the number of cell layers there are, and two, the shape of the cells. So this is really easy. I love this. When we look at the number of layers, we count to 1, and if there's one cell layer thick, then it's simple epithelium. If it's more than one layer, then it's stratified. That's it, 1 or more than 1. So we're going to use simple epithelia, the 1 layer thick, in areas where we need a barrier, but it needs to be really, really, really thin. For example, the air sacs in your lungs. We want blood on one side, and we want air on the other. You don't want your blood in your air because then you're bleeding. So we want a thin barrier, but we also want to diffuse oxygen and carbon dioxide very quickly across that barrier, and that's where simple epithelium works really, really well. Stratified epithelia, on the other hand, can be 2 or more layers of cells. Only the basal layer is in contact with the basement membrane, and we have this in areas that are subject to mechanical stress. So those layers can resist wear and tear, or if they get knocked off or rubbed off or bumped off or something, then because of the high rate of regeneration of epithelial tissue, we can just grow more and cover the area up again. So there is a type of epithelial tissue that doesn't quite fit into our first 2 categories here, and that is pseudostratified epithelium. Pseudo means false, stratified, of course, is layers. So pseudostratified looks like there's multiple layers, but there really isn't. So these are pretty special in the body. We only have them in 1 or 2 locations, and we'll go over those in a little bit. When we look at the shape of epithelial tissue, we're going to look at the shape of the cells. And for purposes of simple epithelia, it's pretty easy. But when we get to stratified epithelia, where we have multiple layers, we're only going to look at the very top layer of epithelial cells, or the apical layer. So squamous means scale. So these are flat and wide, very flat little pancake type cells. Cuboidal looks like a cube. They're about as tall as they are wide. And then columnar cells are long and tall like a column. And then, of course, we have categories that don't quite fit into these 3 here. Transitional epithelia, the cells kind of change shape depending on how much they've been stretched. And they're really, really good at stretching. And so we use these in areas that might need to stretch a little bit, like the bladder or the urethra. So let's look at some qualifications here, some classifications. Here we have a squamous cell. It's nice and flat, little pancake here. So our simple epithelium looks like this, where we've got our simple epithelial layer and then a little bit of a basement membrane on top, and that's it. Stratified epithelia, we look at the epithelial tissue, which is going all the way down to here, and we've got something interesting. It's so thick that if we pour our syrup, and in this case our syrup is coming from the bottom, right? It's the bloodstream down here where it's vascular, and our epithelial tissue is avascular. So our syrup needs to diffuse in this direction, but the problem is it's too thick. And so what we're going to have is these cells are actually going to die and be dead. So we find stratified epithelium, stratified squamous epithelium, because if you look at the apical surface here, these are little flat guys. We find that in our skin. Our cutis, our membrane that covers our body, is made this way. And that way, when you spill stuff on yourself or you scrape a little bit of skin off, it's not really a big deal. Those cells were dead anyhow, and you're continuously growing more of them down in this layer, and the older ones are getting pushed up toward the surface. Well, they'll eventually fluff off. Perhaps you had a temporary tattoo at one point in your life, and it's temporary because they flake off after a while because the ink is only applied to the apical surface of the cutis, and as those cells are dead and can't repair themselves, they start to break off. There's no way to fix them, and they'll take the ink with them as they fall off. So when we look at epithelial tissue, we can have simple epithelia. It could be simple squamous, simple cuboidal, simple columnar, or pseudostratified. And then we can have stratified epithelia, which can be stratified squamous, stratified cuboidal, stratified columnar, or transitional. When we look at our stratified squamous, for example, like in the cutis, some of it can be keratinized, and then some of it's non-keratinized. Keratin is a very tough, durable, waterproof protein that we load into the cells, and the cells get so full of it that they're dead. Kind of like my garage is full of crap, and so it no longer functions to park cars in. The keratinized cells are so full of keratin that they can no longer function as cells, and they're just dead. We also have areas of the cutis that are non-keratinized. And these areas, the skin, or I should say the surface cells, are still alive. They're non-keratinated, but they're still part of that stratified squamous protective layer.